Hello everyone, very warm welcome to this amazing platform of Infinity Learn by Sri Chaitanya. My name is Kiran and I am your biology faculty at Infinity Learn. Students, let's talk about today's session. In this class, I will continue the discussion on previous year questions that have been asked by NTA in NEET examination from 2013 to 2021 related to chapter number 6 of your 12th class NCERT biology textbook, and the chapter name is Molecular Basis of Inheritance. We have already covered or discussed the questions that NTA has asked from 2013 to 2021 related to the introduction, the DNA part and search for the genetic material until replication we covered in the previous class, part 1. This is part 2 where we will discuss the important previous year questions that NTA has asked from transcription till the end of the chapter, that is DNA fingerprinting, right? So, it is going to be an amazing session, right? So, stay connected, and also let me tell you, the objective of this session is not to give you the right answer, because you already know the right answer and it is readily available in several books in the market, right? The objective is little different. What I aim in this class is you should not only understand that which one is the right option, but also the other 3 options why they are wrong, and with the help of questions we are going to discuss, we are going to revise all the important concepts and facts on which NTA has already asked questions, and there is a high probability that NTA can again ask question in your coming NEET 2022. So, the objective is to revise the concept with the help of PYQs. Hello Abhirami, hello Ariman, very warm welcome, and I will really appreciate if you guys are going to participate, right? So, let us start the session. So, here comes the very first question from transcription, right? The first one is, this question, my dear students, was asked in the latest NEET 2021. Latest NEET 2021. So, let me read out the question and then I would really appreciate if you can give me the right answer. So, asking about the correct statement, right? Can you guess which one is the correct statement in this question? Can you guess which one is the right answer according to this question? So, let me tell you, right? Let me read out all the 4 and help you in understanding which one is right and why the other are wrong. So, let me read the first one. Split gene arrangement is characteristic of prokaryotes. No students, it is of eukaryotes. It is of eukaryotes, and let me tell you, my kids, what is split gene arrangement? Arrangement of intron, exon in a gene is, or in this way, when information is stored inside DNA, such arrangement is known as split gene arrangement, where the exons or important parts from which the genetic info or in which genetic information is stored, rather I should say, is interrupted with the presence of introns. Such an arrangement is known as split gene arrangement, and it is seen in eukaryotic genes. So, eukaryotic genes are split gene, right? Next question. In capping, methyl guanosine triphosphate is added to the 3'N of HNRA. No, capping is done on 5'N, right? Tailing is done on 3'N. RNA polymerase binds with the Rho factor to terminate the process of transcription in bacteria. Yes, Rho factor and sigma factor, if you remember students, they provide specificity to RNA polymerase. So, please understand, with the help of sigma factor, RNA polymerase knows, or with the help of sigma factor, it can start or initiate the transcription process in prokaryotes, and with the help of Rho factor, RNA polymerase can cease or terminate or stop the transcription process in prokaryotes, example bacteria. So, this one is right. So, let us see the 4th one also. The coding strand in transcription unit is copied to an mRNA. No, during transcription process, it is not the coding strand but template strand. Template strand is coded or transcribed, right? I hope this much is clear to all of you. Hello everyone. Okay, let us see the next question now then. Name the enzyme that facilitates the opening of the DNA helix during transcription. So students, many a times when you are in hurry during your examination, during your NEET exam, you do develop anxiety, right? And you just run over the question, and at that point you might skip on some very important hints that examiner has provided in the question. And a very important hint in this question is transcription, right? So if you do not pay attention on this one, you might give answer according to replication, because according to replication, during replication, who opens the DNA helix? It is helicase, right? But when it comes to transcription process, right, copying genetic information from DNA to RNA during transcription process, it is actually RNA polymerase only that will help in opening the DNA helix. Please remember this, okay? Very important question. Yeah, match the see, there is column 1, there is column 2, and this column is this. In A column there is 3 types of RNA polymerase, 1, 2 and 3, and with the options you can understand all these are for eukaryotic, right? During the eukaryotic transcription process, it will be, or these 3 will be used, RNA polymerase 1, 2 and 3. Right, now let us see what is the specific function of these RNA polymerase during transcription process in eukaryotic cell. So see students, RNA polymerase is responsible for forming rRNA. RNA polymerase 2 is responsible for forming the primary transcript, right, that is the hnRNA. Next, RNA polymerase 3 will do formation of tRNA. So 1 will do of rRNA, 2 will do of hnRNA, and 3 will do of tRNA. This question was asked in 2019 Odisha NEET also, and in this year also it was asked. But let me tell you, this question in 2021, latest one, wasn't asked exactly like this. Rather, it was not a match the following, rather it was a statement type of question where the question reads that what is the function of RNA polymerase 3 during eukaryotic transcription process. So it was like that. But why I have given only this one? So that in this question that was asked in 2019, you can actually summarize or revise all the functions of all the 3 important RNA polymerase, right, that participate in transcription process in a eukaryotic cell. Next, which is the correct statement, right? So let us try to solve this question. I hope it is clear to all of you till now. Abhi, Rami and Ariman, I would really appreciate if you participate. So try to give the right answer. Doesn't matter your answer is right or wrong right now, because this is a practice session. In a practice session, what really matters is giving it a try. So if you will give it a try, what maximum can happen, right? Either you will get it right, that's going to be amazing, good. But if you get it wrong, I am there to help you, I am there to teach you. So at least give it a try. Hello Sunil, give it a try to this question. Select the correct statement. So let me help you. Which one is the correct one, right? So Franklin Stahl coined the term linkage. No, my lovely kids, linkage term was coined by T.H. Morgan. T.H. Morgan. And Franklin Stahl and Michelson, remember these two guys did experiment on E. coli, right, where they were trying to prove that DNA replicates semi-conservatively. So that was the work by Michelson and Stahl, remember. Next, Punnett square was developed by a British scientist. Yes, this is in fact correct. This is wrong. This is in fact correct. It was by Reginald G. Punnett. The name of the scientist itself include that Punnett word, and it is used during calculating the results of the crosses. It can be monohybrid, dihybrid, trihybrid crosses. Next is spliceosome take part in translation process. You know students, transcription in fact is the process where spliceosomes are used. So, spliceosomes are used during transcription process. Now, let us see what the spliceosomes will do. Spliceosome actually take part in post-transcriptional events. Rather, I should not even say transcription, I should say during post-transcription events. So, what will happen during transcription? During transcription in a eukaryotic cell, there will be formation of a primary transcript, that is hnRNA. hnRNA will include or contain exons and introns. So, during post-transcriptional processing, hnRNA will undergo the post-transcriptional processing where introns will be removed, right? Introns will be removed and then left out exons will be joined. So, spliceosome work is to remove or cut out the introns from hnRNA, right? Next, transduction was discovered by S. Altman. No, this is wrong, students. Transduction was in fact discovered by Zinder and Lederberg, right? And NTA has somehow asked this information again and again. So, try to remember this, by Zinder and Lederberg. Next, I hope you got the right answer. Very interesting question. This one is a very interesting question and I request all of you to participate in this question. Rather, I should say it is a conceptual question. So, let us practice at least, let me tell you, NTA, I have written here 2018, but I guess if I am not wrong, NTA has also asked similar to this question in 2019 also, right? It might be Odisha NEET, okay, 2019. Similar question based on similar concept is asked. So, let us try to understand how to solve such questions. The question says, if this is the sequence from the coding strand of a gene, remember gene, the segment of a gene that participate in transcription process is known as transcription unit, and in a transcription unit there is a promoter, there is a terminator, and in between there is a structural gene. Structural gene has two strands, coding strand and template strand. Remember, template strand participate in the transcription process where genetic information present on the template strand is coded into an RNA, right? So, remember students, the sequence of nucleotide that will be present on an mRNA will be similar to the one that are present on a coding strand, not the template, because mRNA is produced using the sequence, nucleotide sequence that is present on template. Hence, the sequence, nucleotide sequence of mRNA and of the template will be complementary and not same or similar. Rather, it can't be same because RNA is going to have U and DNA strand will have T in place of U. So, remember students, the fact or the concept is the sequence of nucleotide in mRNA or RNA will be similar to the one that is present on a coding strand of a structural gene. Yes, with only one difference, the coding strand, wherever it has T, you have to replace it with U. So, the right answer is this, because see this is the sequence of coding strand. So, the corresponding mRNA will have not exactly same but similar to this, only the difference will be wherever there is T, there will be U. So, A double G, T, A, T. So, in spite of T, they will have U, uracil, A, U, then G, C, G, C, C, G, C, A, and again in place of T it will be U. Simple, very important question, very conceptual, please remember this. Okay, now let us take the next question. Which of the following RNA should be most abundant? This question was asked in NEET 2017, abundant in animal cell. So, let me tell you students, out of the three major type of RNA, tRNA, rRNA and mRNA, the most abundant one is rRNA. Approximately 80 to 85% of the cellular rRNA is made up of or comprised of rRNA. Talking about others, tRNA can be up to 10 to 15%, and mRNA is approximately 5%. mRNA is approximately 5%. Please remember these facts. Next question. Come on students, participate. Next, again 2017, spliceosomes are not found in. Now you must be clear where spliceosomes will be found, because you know the function of spliceosome. They are going to cut and remove interons from the exon part in post-transcriptional processing, right? Processing events that take place in a eukaryotic cell, right? So, spliceosomes won't be found in prokaryotic cell. So, out of the 4 options, which one belongs to prokaryote? Very simple. Bacteria is a prokaryote. Bacteria is a prokaryote, and hence there will be no spliceosome, because spliceosomes are not required in them, because bacterial genes are non-split type. They do not have presence of interons among the exons, right? Next, fungi, animals and plants all are eukaryote. They will have split genes. They will require spliceosome to cut and remove the interons from the exons. Next, this question was asked in NEET second phase of 2016. The equivalent of a structural gene is. Very simple question students. If you remember, NCERT has actually mentioned these 2 terms, gene and cistron, right? So, equivalent term for a structural gene is cistron, because from a cistron a polypeptide chain can be formed. From a cistron a polypeptide chain can be formed, right, during translation process. Polypeptide chain can be formed. Next, see students, cistron. See, now let us see what are the other terms. What is a muton? Muton is actually a small segment or a part of chromosome that can actually undergo mutations, right, and change its sequence of nucleotides. What is operon? You already know operon is a set of arrangement of genes. You remember Lac Operon? Lac Operon has a polycistronic gene ZYA, which is regulated with the help of a common promoter and has regulatory genes, right? That's arrangement is known as operon. Next is what is recon? Recon is the smallest unit or a segment or a very small part of a chromosome that can participate in recombination process. So, I hope students till now all the points are crystal clear to all of you that why other options are wrong, right? I hope it is clear. The next question. Let me know if it is not clear in the chat box. I would love to address your doubts. Next question. Again, question in NEET 2016. Which of the following rRNAs act as a structural RNA as well as ribozyme in bacteria? What is a ribozyme? It is an enzyme. What is the function of an enzyme? This enzyme will do catalysis, right? It is going to catalyze some or the other type of a reaction. So, ribozyme is that type of an enzyme which is biochemically or biochemical nature, or I can say this type of ribozyme is a type of an enzyme that is made up of nucleic acid, right? RNA, rRNA. So, in bacteria 23 srRNA not only act as a structural RNA but also as ribozyme, where it will catalyze peptide bond formation process during translation. 23 srRNA, right? Clear? Good. Next question. NEET 2016. See here students, DNA dependent RNA polymerase catalyze transcription on one strand of the DNA which is called. So, tell me students, during transcription process which strand is transcribed? Template strand or coding strand? Very simple. Template strand is the one that get coded, right? Please remember this. Next. So, I can give you a brief discussion on this part. This is what? This is a transcription unit. This is a transcription unit. And see my kids, in this transcription unit there is promoter, there is structural gene, there is terminator, and in this structural gene there is template strand and there is coding strand, right? This template strand has the polarity 3 to 5, and hence this can participate in transcription process, because remember, this DNA dependent RNA polymerase can transcribe only in one direction, that is 5:3, hence making the strand 3:5 as the template one. Please remember this coding strand, although its name is coding, but is not going to get coded during transcription process. But remember, what is the function? So by default, all the references in a transcription unit are made with the help of or considering the coding strand, right? The coding strand has polarity 5:3, where how you give reference of the promoter. Promoter is present upstream, right? When you say it is upstream towards the 5' and, so who is promoter is present towards 5' and of whom? Of coding strand or of template strand? It is of coding strand. So coding strand का डायरेक्शन यहाँ पर 5' है और प्रमोटर को आप 5' अपस्ट्रीम कहते हैं, इसका मतलब आप प्रमोटर का डायरेक्शन अकॉर्डिंग टू कोडिंग स्ट्रैंड दे रहे हैं, और टर्मिनेटर का डायरेक्शन यू डिफाइन इट डाउनस्ट्रीम टुवर्ड्स 3' and, that means it is the coding strand whose towards 3' and the terminator is present, right? So please remember, references are made with the help of coding strand. I hope this much is clear to all of you. Good. So it was a good revision of the transcription unit with the help of this question, right? And that is why this session is amazing, because we here not only solve the questions, but we also revise all those important facts and concepts on which NTA might ask question in NEET 2022. Next. Very simple, direction of synthesis and direction of reading of the template of DNA strand. Right now I explained to you. What is the direction of the template strand? Template strand is 3:5, right? Remember this. And I told you RNA polymerase, DNA dependent RNA polymerase is going to catalyze only in the direction of 5:3, hence making the strand 3:5:1 template. So direction of RNA synthesis is 5':3' and the template strand is 3':5'. Please remember, right? Next question. This was asked in NEET 2014. Good question. Okay. So now let us see the next one. Yeah, so till now these were the important questions. Almost all the questions I have discussed that NTA has asked from NEET 2021 to 2013 based on transcription topic. Now let us see what are the important questions that NTA has asked from NEET 2021 to 2013 on the topic genetic code. So get ready, some important and interesting questions are coming now. Under which of the following conditions there will be no change in the reading frame of the following mRNA? So, my lovely kids, let me tell you something very important over here is reading frame, right? This question is about reading frame. So, whenever, you remember when I was discussing about the genetic code and mutations, I told you insertion or deletion of one nucleotide or two nucleotide, insertion or deletion of one or two nucleotide will change the reading frame from the point where that particular nucleotide is inserted or deleted. So, wherever a nucleotide is or one or more than one or two nucleotides are added, from that point, after that the reading frame, the way how codons you used to read before mutation and after mutation will change. The codons will change from the point of insertion, right? So, very simple question. It is asking under which of the following condition there will be no change. So, only students when there is insertion or deletion of three nucleotides, consequently, means one after the other. It is not like one here, another here and the third one there, not like that. Consequently, like one after the other, if you add or delete three nucleotides togetherly, that will not change the reading frame from the point where they are either inserted or deleted, right? So, because addition or deletion of three nucleotide or multiples of 3 nucleotides will do what? Either add or delete codons. Either they will add new codons or they will delete some of the existing codons. Hence, the reading frame of the already existing codon won't change, right? So, remember, I have already provided you the answer over here. So, deletion of 3 nucleotides at any position will not change the reading frame from where they are inserted or deleted. So, this is what GGU, this one, they are consequently present one after the other, right? And hence, adding or deleting of 3 or multiples of 3 will not make any difference, right? So, now let us understand the next question, students, here. Let us understand the next question here. So, I hope now you must have got the answer, right? Next question. Insertion of G at fifth position, deletion of G at 5th position, and insertion of A and G at 4th position. All this will lead to frame shift, insertion or deletion, mutation, right? I hope this question is now pretty easy for all of you. Next question. Which of the following features of genetic code does allow bacteria to produce insulin by recombinant DNA technology? Yes, students, I really like this question because this question actually connects 2 chapters, genetic engineering, right, the biotechnology and molecular basis of inheritance. So, beautifully NTA has asked this question, right? So, please try to understand how these 2 things are connected. So, you know, genetic code is nearly universal. Why nearly universal? Because there are some exceptions with respect to mitochondrial codons or in some protists, right? But leaving them, genetic code is nearly universal. That means, for example, U, triple U. For example, U, triple U in my cell, human cell, eukaryotic cell, code for phenylalanine, right? The same genetic code in a bacteria will also code for phenylalanine, and hence because of this property, because of this salient feature of genetic code that is nearly universal, hence we are able to produce or synthesize insulin using genetic engineering inside a prokaryotic cell like bacteria. So, that is the correct answer, right? So, genetic code is nearly universal, hence allowing us to produce insulin in a prokaryotic cell, human insulin in a prokaryotic cell like bacteria using genetic engineering, right? Next question. Yeah, this is a good question. I want everybody to read out this question first and then let us togetherly solve this question. Very good question, right? If there are 999 bases in an RNA, okay, that code for a protein with 333 amino acid. Next, and the base at the position 901 is deleted such that the length of the RNA becomes 9998. How many codons will be altered? So, remember students, this is based again on that same concept I taught you right now, right, a few minutes ago, where I was telling you about the mutations in the genetic code. I told you whenever a single or 1 or 2 nitrogen base is either added or deleted, it will change the reading frame of the codons that are present after that point where this insertion or deletion is done. So, how many codons will be altered? How will you come to know? All those codons that are present just after this 90. See students, for example, I can say 1, 2, 3 and like this there are 999. There are 999 bases and somewhere here 901. This base, this nitrogen base was deleted. The base position at 901 base which was present at 901 or 901 base is deleted. So, after this students, still here there won't be any change in the reading frame. So, the genetic codes codons will hold the same meaning before and after the mutation. But all those codons that are present after this, their reading frame will change. So whatever meaning the genetic codons were holding after this one, before mutation and after mutation will change. So right now what we need to understand is how many codons are there after this 901 position, and only in them the alteration will be seen. So question becomes now pretty simple, pretty easy. So let us see this. How many nitrogen bases are present before 901? Before 901 there will be 900 nitrogen bases, and you know one codon is made up of 3 nitrogen bases. So divided by 3, how many codons will be formed? How many codons will be formed? 3, right? So see students, 300 codons will be present. So 300 codons are present before this deletion is done. And you know how many amino acids have been coded? 333 amino acids are coded. Each codon is going to bring or going to represent to one amino acid, right? Because one particular codon corresponds to one particular amino acid. So from this information you can understand if there are 333 amino acids, that means in that total mRNA there were 333 codons. So the total codons were 333 before mutation. That means deletion of this 901 nitrogen base. Now what you want to do is this, before mutation these many were there, and you know after deleting this, in this section the reading frame will not change. That means 300 codons will not be altered. So 333 minus 333, what are these? These are the codons. These are the codons whose reading frame will not change even after mutation, because reading frame of the codons will change from the point where the insertion or deletion is done. So finally you get 33 codons whose reading frame will change. Simple. Tell me, is this question crystal clear or not? I really hope I tried my level best to simplify this question for all of you. But still, if there is any student who wants me to repeat it or who wants me to explain in any other way, do let me know. So the answer is 33. Okay. So let us take the next question. Which of the following is the starter codon? Which of the following is the starter codon? Starter codon means, my dear students, initiation codon. Initiation codon is the one who is going to initiate or start the translation process, formation of protein or polypeptide chain. So see student, it is in fact AUG. AUG is the initiation codon, starter codon, that has dual function. It will code for methionine as well as it will code for methionine amino acid, as well as it will work as start the translation process, right? Please remember it has dual function. And please remember, with this question itself, we all should be able to understand and revise and remember this fact that these codons UAA, UAG and UGA are stop codons. I repeat myself students, UAA, UAG and UGA are what? They are stop codons. What is the function of stop codons? They will stop or cease or terminate the translation process, right? I hope this much is clear to all of you, my kids. Next question. Which of the following is a property? Which of the following is not? 2013 Karnataka question. Which of the following is not a property of genetic code? Important question with this question. we are going to revise all the salient features of a genetic code. So stay connected and listen carefully. First one, genetic code is non-overlapping. Yes, genetic codes do not overlap. What does that mean? Simply, listen this carefully. How genetic code is non-overlapping? So the particular nitrogen base, a codon is made up of 3 nitrogen base, right? So a particular codon or a particular nitrogen base that has become part of one codon will now not participate or become part of another codon. So simply, for example, if it is like A, U, A, I can write G or G, C, A. So if this is what, hypothetical example, if this is what one codon, if this is one codon and this is what the second codon. So see students, second codon. So this A, if this A has become part of this codon, this A will not participate, and become part of second codon also. That means codons or nitrogen bases present in the codons do not overlap. If these 3 are present in one codon, that they will not become part of the second one. Hence, genetic code is non-overlapping. Second, genetic code is ambiguous. No, genetic code is not ambiguous, and this becomes the answer to this question. What does ambiguity means? Very simple. Generally students get confused between these two, ambiguity and degeneracy. So right now I want everybody to understand these two important points. So see my kids, what is ambiguity and what is degeneracy? Ambiguity means that, let me tell you what is degeneracy means first. So degeneracy means amino acids, right? More than one codon can code for the same amino acid. That is degeneracy, right? But one codon will not code at the same time for 2 amino acids. That is ambiguity. Let me at least write it and explain you, because again you might get confused. Just give me a moment. Let me write it and explain you. Just stay connected. Okay, so for example, this is an amino acid, for example valine, right? This is an example of one amino acid, right? So I have codon 1, I have codon 2, I have codon 3, I have codon 4. So I am just giving you an hypothetical example. So one codon will code to one amino acid. One codon will code to one amino acid only. So at the same time, this codon cannot code for valine and also for alanine, right? Right? So what I want phenylalanine. So what I want to make a point over here is one codon at a time will code for one amino acid only. The same codon cannot code for 2 amino acid. So if one codon codes for 2 amino acid, then codon becomes ambiguous, and such property is not seen in the genetic code, and hence the genetic codes are not ambiguous. So this is what ambiguity. This is what ambiguity. But this thing is in fact not seen in the genetic code. But what is degeneracy? Simple. Codon 2 can also code for valine, codon 3 can also code for valine, codon 4 can also code for valine. So more than one codons coding for the same amino acid is seen in genetic code, and this property of genetic code is termed as degeneracy. But this one coding to 2 won't happen. That is ambiguity, and that is not seen in genetic code. So I really hope now you will never ever going to get confused between these two terms, ambiguity and degeneracy. Not only till your NEET exam, but also beyond that you won't forget. I can assure you not. But if you if you were listening to me carefully. Only in that case. If you haven't, please go back and listen this again. How genetic code is universal? Genetic code is in fact nearly universal, right? It is nearly universal. That means the same triple U, if it is coding for phenylalanine in human cell or eukaryotic cell, it will code for same phenylalanine in the bacteria also. We have already done the discussion on this, so I'm not going to repeat it again. Next question. So these are the questions that NTA has asked from 2013 to 2021 based on genetic code topic. Now let us take quickly the questions that NTA has asked from translation topic. See here students, the first phase of translation is very simple. What is translation process? When the genetic information is copied from mRNA to the polypeptide chain, right? So during the translation process, amino acylation of tRNA takes place first. So during the first phase of this translation process, amino acylation of tRNA takes place. Let me throw some light on this process, students. What is amino acylation of tRNA? So during amino acylation of tRNA, what will happen? An activated amino acid will be covalently bonded on this cognate tRNA. This is known as amino acylation of tRNA or charging of tRNA also. So amino acylation of tRNA or charging of tRNA. And what happens during this process? Activated amino acid will be covalently bonded with its cognate tRNA, right? And then this amino acylated tRNA will be ready to do translation process. Next question. And see, this is what I have already explained. See my lovely students, I have already explained this entire process of translation in extreme detail in my series, right? When I was teaching this chapter, molecular basis of inheritance. So if till now, or even explaining you right now, this whole process, this whole explanation of this question, you are still little bit confused or not clear on how exactly the translation process is going to proceed, I request you to go back to the playlist of botany and check where I have taught this topic translation. I can assure you, you will never ever get confused on any concept related to translation process if you check out that video. So do consider checking it out if you haven't done till now. Next question. So here exactly what is happening. This is the tRNA. This tRNA is getting covalently bonded to this activated amino acid, and this is what? This is an amino acylated or charged tRNA that will participate now in the translation process. Next question, students. Many ribosomes may associate in a single mRNA to form multiple copies of a polypeptide simultaneously. Such a string of ribosomes are termed as, so see students, till now NTA has asked these questions more than 2 times, right? More than 2 times the same question has been repeated. So this question has a very high frequency of getting repeated, and you know, even the language is not changed, like almost similar language, and again this question is asked. So please try to understand, such a structure is known as polysome. Such a structure is known as polysome, and this is how it looks like. On an mRNA, there are several ribosomes participating in the translation process. Very simple. Next question. Yeah, so now we'll do questions on regulation of gene expression. Very important conceptual topic. Let us see what are the important questions that NTA has asked till now from this part. Very simple. Which of the following genes of the Lac Operon with their respective products? So you remember, student, this Z, A and Y, this represent to the polycistronic gene of the Lac Operon, right? And this I gene is in fact the regulatory gene, right? The inhibitory gene, the regulatory gene. In a Lac Operon, this I gene was forming a repressor mRNA through which a repressor protein was getting formed, and this repressor protein used to bind to the operator region, because of which RNA polymerase could not bind to the promoter, common promoter of this polycistronic ZYA gene, and hence there was no transcription, right, in the absence of inducer. Remember? So, this I gene will code for repressor, Z will do for beta galactosidase, A will for transacetylase, and Y for permease, right? I'm sorry. So, this is how it used to happen, right? But I want to tell you something very important over here, since this question has been asked. So, let us take a few more minutes and revise all the important concepts related to this Lac Operon. So, see students, which condition I am taking? In presence of inducer. So, remember, Lac Operon needs to be induced. So, in presence of inducer, this Lac Operon will get switched on. Switch on means start of the transcription process of this polycistronic gene ZYA. So, when there will be presence of inducer, Lac Operon will get switched on, and there will be transcription of this polycistronic gene, right? And during transcription process, from this part a Lac mRNA, from this part ZYA polycistronic gene, Lac mRNA will be produced. I really hope students you can see that, right? And as I told you, there is what? There is a presence of inducer. So, who is the inducer for a Lac Operon that can switch on this operon? Simple. The inducer is either lactose or allolactose, which is an isomer of lactose, right? And also beta galactosides can also do that, right? Next students, if there is a presence of inducer, this repressor protein which is formed from the repressor mRNA, and that is formed from this I gene, this regulatory gene, the inducer will go and bind to this repressor protein, because of which the repressor protein will become inactive, and as a result this repressor protein will not bind to the operator region, and hence RNA polymerase will be able to bind to the common promoter of this polycistronic gene and do the transcription process of ZYA, and as a result there will be formation of Lac mRNA, and during translation process from this Z gene there will be formation of a protein that will fold and become an enzyme known as beta galactosidase. What is the function of beta galactosidase? It will help in hydrolysis of the lactose, where it will break this beta galactosidase. Who is the substrate for it? Lactose. It will do the hydrolysis of lactose, help in hydrolysis of the lactose, and as a result glucose and galactose will be produced. What is the function of permease? Permease. So, from this Y gene there will be formation of a protein that will fold, right, and form finally an enzyme known as permease that will help in increasing the permeability of the E. coli or bacteria, so that lactose can enter inside the cell, right? It is or other than lactose, it will also help in increasing the permeability for beta galactosides also. Next, both can act as an inducer for this operon. Next is from this A gene, there will be formation of a protein that will fold and finally convert or form an enzyme known as transacetylase. So, till now whatever function has been discovered by scientists for this transacetylase is this will actually help in modifying the toxic beta galactosides which need to be removed from the cell. That's what a Lac Operon is in the presence of inducer. I hope I have literally helped you in revising all the important facts and concepts related to this Lac Operon with the help of this simple question. Next question, students. Select the correct match. So, let me tell you, it is quite simple. Elliot Jeffrey, if you notice students, he in fact discovered or he invented DNA fingerprinting, right? So, he is not concerned with Streptococcus pneumoniae. Streptococcus pneumoniae, in fact, the work on Streptococcus pneumoniae was done by Griffith, and during his experiment he discovered the transforming principle. Next, Hershey and Chase. On TM we know Hershey and Chase worked on bacteriophages. They worked on bacteriophages where they gave the unequivocal proof that DNA is the genetic material. Messelson and Stahl, they in fact worked on E. coli and not on Polysome sativum. They worked on E. coli and they proved that DNA replicates semi-conservatively. Jacob and Monod, Lac Operon, yes, they did work on Lac Operon. So this is the right option. Now let us see the next question, my kids. Which of the following are part of an operon except right now we discussed right now. So an operator is a part, structural gene is a part, promoter is a part, but enhancer is not a part. So what is the function of operator? On operator only, that repressor protein used to bind and because of which the operator used to affect the activity of RNA polymerase on promoter, right? Next. Question was asked in NEET 2019, very simple, from this Human Genome Project, extremely simple fact-based question. Expressed sequence tag refers to, it simply refers to those sequences which express itself in form of protein. So, gene expressed as RNA and from that RNA later on the processing can be done, the further process can take place. So, what are the expressed sequence tags refers to? The genes that are expressed as RNA. Next. Identify the correct order of organization of genetic material from largest to smallest. Very simple. It will be first one. How? The largest is the human genome, right? Human genome is the largest one. Remember how many nuclear nitrogen bases are there? Remember 10 raised to the power 9. It is quite a large molecule, right? Then it comes to chromosome, then it comes to gene, then it comes to nucleotide. So, nucleotides are made up of gene, gene is made up of chromosome, chromosome is made up of genome, hence making the chromosome the largest one. Very simple question. Next, students, satellite DNA is important. Yes, satellite DNA is important because it shows high degree of polymorphism in population and also the same degree of polymorphism in an individual which is inheritable from the parents to children, and it forms and it helps in doing what? DNA fingerprinting, right? So, the same polymorphism will see in all the cells of the same individual but won't be same in the another one. Next, and they are inheritable. One of the most frequently used techniques in DNA fingerprinting is VNTR. Right now we discussed in the previous one. Next. Yeah, that's it. We have come to the end of this session, students. Okay. Is there any doubt? Please let me know. Okay, that's all then from this session, students. I really hope that everyone enjoyed this session. We discussed and revised all important concepts on which NTA might ask question in NEET 2022 based on the chapter 6 molecular basis of inheritance. So that's all from this class. Give me a thumbs up if you like me and this session, and do not forget to subscribe Infinity Learn by Sri Chaitanya YouTube channel, and also hit the bell icon so that you get notified every time I come live and you won't miss any single session in future. That's all, students. Thank you. Bye-bye.